We all know that with the right kind of help, a child who's really struggling with reading can totally blossom into a fluent reader. But have you ever stopped to wonder what's actually going on inside their brain to make that happen? Today, we're jumping into a fantastic scientific detective story. We're going to explore how targeted teaching can literally rewire the brain of someone with a reading disability. Okay, first, let's just get a sense of the scale here. For at least 9%, and that's almost 1 in every 10 school kids, learning to read isn't just a little tough. It's a huge, ongoing struggle. This is what we call a reading disability, or dyslexia. And right there, that's our central mystery. See, we've got mountains of evidence showing that the right interventions can make a massive difference in reading skills. But what does that change actually look like on a neurological level? How can teaching something like phonics physically change the most complex object in the entire universe? That's the question scientists were determined to answer. To solve any mystery, you first have to look at the clues you already have. And in this case, that means understanding the neurological foundation of a reading disability. So here's what we see at a network level. In a typical reader, there's this really rich, coordinated dance of activity in the brain's left hemisphere whenever they're processing words. But for folks with a reading disability, that whole network is just quieter. It's consistently less active. It's almost like the communication lines for reading just aren't fully hooked up. But, and this is so important, this neurological challenge is not a life sentence. Not at all. It's just a starting point. We know that specialized, super explicit, phonics-based instruction is the gold standard. It's been proven again and again to help people learn to read well. Why? Because the brain is plastic. It can change. So how do you figure out what's really happening in there? Well, a team of researchers decided they were going to crack this case by sifting through decades of scientific evidence, looking for one consistent pattern. Their approach was, to put it mildly, massive. And this flowchart from their paper just shows you the incredible scale of what they did. I mean, look at this. They started with a pool of over 45,000 potential studies. Then, through this super rigorous filtering process, they whittled it down and down and down until they were left with just the 39 most relevant articles to review. That is an unbelievable amount of work. And the clues they were hunting for came in a few different flavors. You've got fMRI, which basically shows you which parts of the brain light up when you're reading. Then there's functional connectivity, which is all about how those different regions talk to each other. And finally, you have structural imaging, which looks for actual physical changes in the brain's wiring. Okay, so with all these clues gathered, it was time to find the smoking gun. The researchers used this really powerful technique called a meta-analysis. It's where you combine the data from a bunch of the strongest studies to see if one single consistent pattern of brain change finally emerges. For this, the real core of their investigation, they zoomed in on 8 key studies. What was special about these? They all had fMRI scans from both before and after an intervention. This gave them a really clear before and after picture for 151 different people. The goal was simple: find that one change that showed up again and again and again. Now, going into this, scientists pretty much had 2 main theories about what was going on. The first was the normalization hypothesis. The idea here is that the intervention basically fixes the original reading network, getting it to work the way it's supposed to. The other theory was compensation, that the brain just says, okay, this route is blocked, and builds a whole new detour, often using parts of the right hemisphere to get the job done. So, which one was it? This was it, the million-dollar question. After all that work, all that data, what was the one consistent, statistically significant change that reading instruction created inside the brain? Well, the answer was not what anyone was expecting. It was a twist in the story that kind of changed everything. That's right, nothing. The big meta-analysis, the most powerful part of their whole investigation, it came up empty. There wasn't a single brain region that consistently changed across all of the studies. No smoking gun. So, did the case just go cold? Was the whole thing a failure? Absolutely not. This surprising result, this lack of a simple answer, was actually the most important clue of all. It told them they were asking the wrong question. The answer wasn't a simple one-size-fits-all fix. It was something way deeper and way more interesting about how our brains learn. The real secret here isn't about fixing one broken part or building one specific detour. It's about something way more dynamic and way more personal. The researchers themselves put it perfectly. This whole idea of normalization versus compensation, it's just too simple. It's a false choice. The reality is much messier and, honestly, much cooler. We need a whole new way of looking at this. And the key to this new way of thinking is a concept called functional connectivity. So instead of just focusing on which individual brain areas light up, functional connectivity looks at how they all talk to each other. Think of it like a sports team. The intervention isn't just changing one player, it's changing how the entire team communicates and works together. The brain's strategy is this incredible dynamic rewiring. It's fine-tuning the whole network. And that can mean boosting some connections to make a pathway stronger, but, and this is wild, it can also mean weakening other connections to get rid of distracting noise and make the whole process more efficient. It even involves physically strengthening the white matter, those information highways that connect different brain regions. Now, this new, more complicated picture also helps explain why the scientific evidence has been so, well, all over the place. Because when you look really closely at the research itself, you start to see some pretty big gaps in how these studies have been done over the years. Take a look at this chart. It's from the paper's own quality check of the 39 studies. Green means a study did a good job, but just look at all that red, yellow, and especially the blue, which means the info wasn't even reported. For example, look at that domain 12 row. Almost none of the studies had a big enough sample size. This shows us pretty clearly that the science here has some real weaknesses that need to be addressed. And here's maybe the biggest weakness of all. A whopping 70% of the studies they looked at did not use the gold standard for this kind of research. Only 30% actually compared the kids getting the intervention to a control group of kids who also had a reading disability but didn't get that specific intervention. And why is that such a big deal? Well, think about it. Without that control group, you have no way of knowing if the changes you're seeing in the brain happened because of your awesome teaching method, or if they're just changes that would have happened anyway, as the child's brain naturally developed over time. It makes it almost impossible to prove cause and effect. So all of this leaves us with this really profound and exciting idea. Maybe there isn't one single way the brain changes, because every brain finds its own unique path to becoming a reader. And that leaves us with this final incredible question for the future. If every brain solution is different, can we one day use this knowledge to design a truly personalized, unique intervention for every single child?